My name is Morgan Robinson and this is my video for Exam 2. Today we will be learning about epistasis. Epistasis is when the expression of alleles at one locus covers up or changes the expression of other alleles at another locus. Due to this, multiple different genotypes can lead to the same phenotype. As you can see in the chart, there are actually 6 different types of epistasis. Today we will only be looking at one, which will be single recessive epistasis. Single recessive epistasis is when the homozygous recessive condition at one locus masks or modifies the expression of alleles at the second locus. Homozygous recessive is when the organism has 2 copies of the recessive allele. For example, the genotype would look like a. To apply and further explain epistasis, and specifically single recessive epistasis, we will be using a pathway to explain fox coat color. For this pathway, we will start with the white fox, which can also be known as the colorless precursor. In order to move on to the brown intermediate color, enzyme A will need to be present. However, if the fox is homozygous recessive in the A loci with 2 little a's, enzyme A will not be present, and the fox will stay white. If the fox ends up not being homozygous recessive at the A loci, they will be able to move on to the brown intermediate color. However, they will then have to go through the same process on the B loci in order to move on to the black color. Enzyme B will need to be present. However, just like before, if the fox is homozygous recessive in the B loci with 2 little b's, enzyme B will not be present and the fox will stay brown. If the fox is not homozygous recessive at the B loci, they will move on to the black color. To further explain our epistatic pathway, we will use examples of crosses to show how the proportioning of phenotypes from our genotypes works. To do this, we will use a dihybrid cross. First, we will be doing a cross of the genotypes big A, big B, little b, and big A, big B, little b, which will be a cross between 2 black foxes. We will then set up a Punnett square to illustrate the cross, and our results will show this. As we can see, the cross gives us a genotypic ratio of 3 to 3 to 1 to 9. Anything with the genotype of big A, big B, will give us a black phenotype, and anything with the genotype of big A, little b will give us a brown phenotype. For white, there will be 2 different possible genotypes of a, big B, and a, little b. Although there are 4 genotypes, because 2 of them code for white, we add them together and end with a phenotypic ratio of 4 white, 3 brown, and 9 black. Now we will do another dihybrid cross using the genotypes big A, big B, and a, big B, little b. This will be a cross between a white and black fox. Again, we will set up our Punnett square and our results will show the genotypic ratio of 8 to 0 to 8. Anything with the genotype of big A, big B will give us a black phenotype. Anything with the genotype of a will give us a white phenotype. Usually anything with a big A and 2 little b's as the genotype would give us a brown fox. However, there are no genotypes for brown seen in this cross, so our phenotypic ratio is 8 to 0 to 8 with 8 black, 0 brown, and 8 white colored foxes.